All right. Hi, everyone. I am Liz Sorani, the Director of Research at FARA. Welcome to the FARA Flash Talks. For already 5 years now, FARA has been celebrating FA Awareness Month by inviting junior investigators to present their research to our community. This year, we have asked the grad students who received the fellowship from FARA and the postdocs who received the FARA Fellow Award to present at our Flash Talk series. Both these programs, the fellowship, the graduate fellowship, and the FARA Fellow Award were started just last year. So the investigators that you'll hear from today are the very first recipients of both awards. As for previous years, we are asking them to present their finding in just 5 minutes and one slide, and to explain their research in simple terms that can be understood by anybody who doesn't have a science background. So let's take this opportunity to ask them questions and learn more about what they do and why they do it. And you can type your questions in the Q&A box, and we'll read them after each talk. We are recording the presentations, and we will post them on YouTube. So here are the speakers for today, but before we start, I wanted to provide a quick background, maybe a sort of a glossary to help you understand what our junior researchers will talk about today. So you will hear about GAA repeats. I know you're probably all familiar with your GAA repeat number. You all know that this is the genetic test for FA. GAA are a short sequence of DNA. DNA is your genetic code that is repeated many, many times in the frataxin gene in people with FA. We call it repeat expansion. And you'll hear from Morgan today how this expansion occurred in our ancestors thousands of years ago, and how she's able to trace it in various populations. Then you'll hear about something called epigenetic code. So we just talked about the genetic code. This is what makes every person unique. So the epigenetic code is a code on top of the genetic code. It's like a secret code, for example, in a book, if you read every third word, you can get a secret message. And the epigenetic code is something like that. And it too influences who we are and makes us unique. And you'll hear from Xiaonan how the epigenetic code influences the expression of frataxin and how it can be manipulated and changed to make more frataxin in the cell. Then you'll hear about frataxin itself, this very important protein that is whose levels are reduced in FA. You'll hear about what it does and how it's essential for an assembly line inside the cell that makes component of proteins. So think about frataxin as part of the assembly line that makes batteries inside the cell. Without these batteries, a lot of the cell components, the electronics of the cell, do not work. And you'll hear from Angela how she's trying to identify these electronics, these components in the cells that need the batteries that frataxin makes. And the goal is to identify these faulty components and hopefully fix them. And finally, you hear about point mutations. I mentioned earlier that you all likely know your GAA number, and most people with FA have 2 GAA repeat expansion, one from dad and one from mom, but about 4% have 1 GAA expansion and 1 point mutation, which is a small change in just one letter of your DNA code. It's small, but it's enough to cause low level of frataxin. And you'll hear from Pure how he has found a way to try to fix that using something called oligonucleotides. What is an oligonucleotide, or oligo for short? It's a small piece of DNA made of the same components that make up your genes. And its important property is that it is able to find its precise match in the cell, in this case, the product of the frataxin gene with a point mutation. And this union, this match, fixes the mistake and increases level of frataxin. So remember these terms when we listen to the presentations. And we have two members of our community here with me today, Michaela Holm and David Van Ock, who will moderate the session with the help of Kelly Matten, the patient engagement manager at FARA. And I will just turn it over to them to take it from here.
All right. Hi, everyone. My name is Michaela, and I was diagnosed at the FA about 9 years ago. And now I am in the FARA Ambassador Program and happy to be here moderating some great research. So, for the first flash talk we have here is Morgan Tackett, and she is going to be talking about her project researching why the GAA repeat expansion that causes FA has evolved in some populations but not others.
Hello, just 1 second while I. So can everybody see my screen?
Yeah, looks good.
Awesome. So thank you for that introduction. My name is Morgan Tackett. As you said, I'm a third-year PhD student in Dr. Sanjay Bidichandani's lab at the University of Oklahoma Health Science Center. And just before I begin, I want to thank FARA for just this opportunity to share our research, as well as supporting our research with the graduate research fellowship that was awarded to me last year. So, as many of us know, FA is not seen equally around the world. It affects people from Europe and South Asia, and it's not seen in sub-Saharan Africans, East Asians, or Native Americans. And the purpose of my project is to determine why it is that FA is seen in some populations and not other populations. So FA is caused by an expanded GAA repeat in intron 1 of the frataxin gene. People who have FA, most of them have 2 copies of an expanded allele that have over 100 repeats. And people who don't have FA either have a short normal allele that has 6 to 11 GAAs, or a long normal allele that has between 12 and 30 repeats. And previously, scientists have found that there's a variety of genetic backgrounds on short normal alleles, and long normal and expanded alleles share one main genetic background. And that has led to this hypothesis that once in human history, a short normal allele got a little bit longer to make a long normal allele, and expanded alleles then developed from long normal alleles. And if we look around the world, we see short normal alleles are in every population, and FA is caused by E alleles, so we only see expanded alleles in susceptible populations. But long normals are interesting because we see them in susceptible populations, but we also see them in sub-Saharan Africa. And this is interesting because why didn't these long normal alleles in sub-Saharan Africa go on to make expanded alleles like they did in susceptible populations? This led us to a hypothesis that long normal alleles in sub-Saharan Africa were different somehow than those in susceptible populations. So how we're going to test that hypothesis is we're going to sequence many people who have FA, so many expanded alleles, as well as analyze many long normal alleles from both Europe and South Asia, as well as sub-Saharan Africa, to see if there's any differences. And when we did this, we found that there's 2 main genetic signatures of expanded alleles. We have this blue signature and the green signature of these expanded alleles. And when we looked for those signatures in susceptible populations, you can see there's a proportion of long normal alleles in susceptible populations that have those same genetic signatures. But when we look at sub-Saharan Africa, we see that no long normal allele has those signatures. So this shows us two things. One, it shows us that only a subset of long normal alleles went on to make expansions. This happened in susceptible populations, but it also shows us that it occurred at least twice in history. And we then wanted to know if there's a difference in the GAA length between these blue and green signatures versus the long normals in sub-Saharan Africa. So we took DNA and we measured the GAA, and we found that the green and blue signatures had all over 20 repeats. Okay? And in sub-Saharan Africa, no long normal allele exceeded 20. We then wanted to confirm that with a larger number of samples. So we took over the DNA sequence of over 1,000 Africans and African Americans. And with a little bit of computer programming, we were able to analyze the frataxin gene. And you can see here is GAA repeats and prevalence. And here are our long normal alleles in this population. And you can see that they never go past 20 repeats. So all of this data together refines this model of the evolution of the GAA repeat. And it says that once in human history, a short normal allele became a long normal allele. This happened in sub-Saharan Africa. And it was only after people left sub-Saharan Africa that a small proportion of the long normal alleles got a little bit longer to become what we're calling a proto-mutation allele. That's the long normal alleles that are over 20 repeats. And it's only this step that was able to go on and expand into our expanded alleles that cause FA. We then wanted to know when and where did this process actually happen? So we tapped into this new field of study called ancient genetics. So on this map, you can see people who lived over 5,000 years ago. And anywhere that you see a blue dot is a short normal allele. Anywhere you see an orange dot is a long normal allele. And any of these green stars are somebody who has a proto-mutation allele. So they have the blue or the green genetic signatures. And you can see these proto-mutation alleles have been in susceptible populations for thousands of years, and we don't see them in East Asia or sub-Saharan Africa, explaining why those populations don't get FA. And the oldest sample that we have of these proto-mutation alleles is 9,000 years ago in Sweden and Turkey. So FA has been around or susceptibility has been around for quite a long time. And altogether, this data shows us a story of the evolution of FA as well as when and where it's occurred. But we have a lot more ancient DNAs to look through and to analyze. So we'll get even a better picture of when this occurred as my data finishes. So thank you so much. With that, I would love to take any questions.
Michaela, there's already one question. Do you see it in the chat?
I think it was given before Morgan's presentation.
Sure, we can ask. The question is, do you repeat change over time as FA progresses?
Sorry. So throughout history, that the short normal to long normal, that did change the GA length. And then, as we said, the proto-mutations have a longer GA length, but they don't cause FA. But what is probably being asked is that does the expanded alleles, do they have a different repeat over time? And it does make sense that the first expanded allele throughout history would have been shorter and gotten longer and longer over time as it through generations. But we have looked at the GA length and age of onset and frataxin levels and so on between these two genetic signatures, the green and the blue genetic signatures. And we don't see a significant difference between the two in GA length.
I think also the question was as you know, if it during the life of a person, the GA repeat changes, and we have some data showing that it does. In certain tissues, like the blood is very, very slow. It's like maybe one repeat a year or something like that. You know, it takes a long time. But in certain tissues, we think that that can happen. Obviously, tissues like the brain we don't have access to, so we cannot know for sure, but there is some data pointing at the fact that over the course of the life of a person, yes, the repeat length can change.
Another question is, so are patients that live in Australia or Brazil just from migration?
Yes. So I should have been more clear on this, that it's the original people from Europe and South Asia and the Middle East that get FA. So we see FA quite often in the USA and Brazil, Australia, so on, but that's all due to ancestral migration to those places. So native Brazilians or Native Americans are not getting FA.
And another question I see here is the molecular mechanism that gave rise to the expansion known unequal crossover?
So the specifically the short normal to long normal transition, we don't know exactly what caused that. Some people hypothesize that it was a duplication, actually, but it very well could be an unequal crossover between those. And from long normal, just getting from 12 to 30, which includes the proto-mutation alleles, this likely is genetic stuttering, that you just add one or two GAAs at a time. But that also very well could have been caused by genetic crossover, just unequally, like grabbing one or two at a time. But then the expansion occurs because it's an unstable repeat. So it hyper expands into an expanded allele.
Next, are there any thoughts around environmental factors in certain areas that may have impacted the repeats?
Yeah, that's a really good question. So we can see that there's certain environmental factors that make sense as to why people didn't migrate, the same populations didn't migrate to other populations. So even like sub-Saharan Africa is separated by the Sahara Desert from North Africa, which explains why these populations haven't mixed all that much, as well as mountain ranges and other environmental factors explaining why the same group of people that brought FA susceptibility to where we see it didn't bring it to East Asia. But as far as like evolution in the sense of natural selection, we don't have data right now showing that that has been an influence in it. Right now, our data just show that just migration history of people is the primary influencers of who gets FA and who doesn't get FA.
Quick question for me, Morgan. Is there some point in time where you can pinpoint where it's all started? Because now you had plus 5,000 years ago, but somewhere in evolution.
Yeah, absolutely. So we are consistently getting new ancient DNAs. And how scientists give or get this ancient DNA is they actually get bones of people who have passed away and get DNA from those bones and sequence it and then make it available to other scientists. So as they're getting these remains and the DNA and sequencing it, it takes time for that process to occur. So we are consistently getting new samples, and we're hoping that it will really refine the age of when this is occurring. So right now, the oldest that we can say is 9,000 years ago, but the oldest long normal that we've seen is 38,000 years ago. So we're hoping with more samples, we can get a better estimate of that age of each allele.
Maybe we should move on to the next speaker.
Thank you guys all so much.
Sounds good. Yeah, I can introduce the next speaker, which will be Angela Zhang from Boston College. And Angela is researching the impact of low levels of frataxin on different iron sulfur proteins that can be found throughout the cell.
Thank you. Let me share my screen. Can everyone see okay?
Yep.
Okay.
Do you want to put it in? Yeah, right. Perfect.
Hi, everyone. My name is Angela, and I am at Boston College in the Department of Chemistry, working as a graduate student. And I wanted to thank FARA, Liz, and the moderators for putting together this webinar. It's also a great opportunity for the researchers themselves to learn about the different branches of FARA research. For me, I am interested in understanding the effects of losing the frataxin protein that would have on humans. And this is because, as we're familiar, the loss of frataxin is linked to the development of Friedreich's ataxia, where in patients, there's only about 30% of the necessary abundance as compared to an unaffected individual. And this will have certain physiological consequences because frataxin has been proposed to help in the production of these iron sulfur clusters, which are essentially biology's helper molecules that are made of iron atoms and sulfur atoms. And when they come together into this cluster-like structure, they can then be inserted into proteins and help with the proper function and biological activity of these proteins. And the proteins that contain these clusters, we call them iron sulfur proteins. In humans, there are about 70 or more iron sulfur proteins. And given the link between frataxin's role and the development of Friedreich's ataxia, many previous studies have been interested in understanding how does losing frataxin affect the activity of these iron sulfur proteins. And researchers have found that frataxin depletion in FRDA leads to a loss of activity in a handful of iron sulfur proteins that are relatively well understood model proteins. But how does losing frataxin affect the remainder of the 70-something iron sulfur proteins in humans? It's still relatively not well understood. So that becomes the goal of my research, which is to extend our current understanding on the effects of frataxin loss to the remainder of the iron sulfur proteins found in humans. And one of the ultimate questions that I would like to answer is, does losing frataxin affect the iron sulfur proteins in humans globally, where most of these iron sulfur proteins lose their clusters? Or does it only affect a handful of iron sulfur proteins or a subgroup of these? And the reason I'm interested in understanding this question is because those iron sulfur proteins that are affected by the loss of frataxin and lose their clusters can serve as potential therapeutic targets. And answering this question will also help us further elucidate the exact molecular role of frataxin, which is currently not very well understood. And lastly, connect this role to the mechanism for Friedreich's ataxia's development. So how do we go about to specifically identify those iron sulfur proteins that lose their clusters and their activity in Friedreich's ataxia? We do so by working with a human cell line that is deficient in the frataxin protein and treating them with a reactive chemical that is actually unable to react with any functional iron sulfur proteins that already contain a cluster, but instead can react with disrupted iron sulfur proteins that no longer have a cluster. And using downstream chemical tools, we can then kind of fish out these disrupted iron sulfur proteins and identify them with an instrument called the mass spectrometer. This way, when we identify those disrupted iron sulfur proteins, we can then evaluate them for their potential as therapeutic targets and further our understanding on the role of frataxin in the development of FRDA. So currently, I am still working to finalize the identification of these disrupted iron sulfur proteins, and I would be happy to share with you guys my results when I find out more about them. And I'll take any questions. Should I answer this one that I see?
I'm not really sure. I think this was from the previous talk.
Oh, okay.
I don't see any questions, but maybe, Angela, would you talk a little bit more about what we currently know about the proteins that we know have iron sulfur clusters and how they affect all the dysfunction in the cell?
Yes. So iron sulfur proteins, they play very diverse biological roles in many kingdoms of biology, not just humans. They can help with the production of energy, such as making ATP in the mitochondria. They can also help with replication of our genome, replicating DNA during cell division, as well as just metabolizing and breaking down nutrients that we consume. So their role is very diverse. Yeah.
Yeah.
I see one question.
I think it's in the chat, right?
Right.
There's one in the chat.
So yeah, the question is, which type of mutant cells will you employ for this study? This is a cell line that I constructed myself using a human cell line. I deleted the frataxin gene and verified that there's very, very low levels of the frataxin protein in the cell. And we decided to use this to mimic the disease environment of Friedreich's ataxia and perform our further studies.
There's another one. Michaela, do you see it?
Yes. Is it possible that the disruption of the iron sulfur clusters may happen as a cascade? So, for instance, frataxin makes some loose iron sulfur, then other proteins make yet others lose iron sulfur, and etc.
It is possible, depending on the molecular, like what our understanding of the molecular role of frataxin is. If frataxin is actually helping with the assembly of the clusters, then this will be a more global effect. Versus if some iron sulfur proteins, if they serve as chaperones or target proteins, then yes, if they don't receive the clusters from their upstream iron sulfur protein, then whatever their target is meant to receive this cluster will not be receiving that cluster. But if, for example, a conatase does not present its iron sulfur cluster, it won't cause another iron sulfur protein to lose their cluster because that is not a conatase's role.
Next is, how healthy are these frataxin deficient cells? Are there special growth requirements?
So we do see that there's no special growth requirement. I would grow them in the same conditions as the wild type frataxin presenting cells. But we do see that these proliferate visibly slower than the wild type cells, maybe at 60 to 70% of the doubling rate as just wild type cells.
Okay. Let's move on. Thank you, Angela.
Thank you.
Hey, let me take over the moderation here. I'm David van Och, the founder of the nonprofit organization UFO2 based in Belgium. Now, UFO2 is an organization my wife and I started when our two youngest sons, currently 9 and 14 years old, were diagnosed with Friedreich ataxia around 2.5 years ago. With UFO2, we financially support FARA research as best as we can. Now, with this, I would like to introduce Xiaonan Zhuang from Columbia University. Xiaonan is researching epigenetic changes or DNA modifications that might affect how much protein the FXN gene can produce. Xiaonan, floor is yours. You're on mute.
Xiaonan, we can't. Oh, there, we hear you now. Yeah.
Oh, 1 second, sorry. Okay, thanks for the introduction. Hello, everyone. My name is Xiaonan. I'm postdoc at Columbia University, and my mentor is Dr. Shan Liu. Today, I'm very happy to share our work progress on the Friedreich ataxia. It's a project collaborative with Dr. Giovanni Dinuti's lab at Columbia University, and also I want to appreciate the FARA support to our project. As we know, the Friedreich ataxia is caused by the depletion of frataxin. So in our project, we are asking, could we rescue FXN expression through the epigenetic layer? So what is epigenetic? I believe you already have some basic concept from this introduction. Let me further explain it. Epigenetic is how the cell regulates the gene expression without affect the gene sequence. As you can see here, there are lollipop on the DNA, we call it DNA methylation. And also there are some chemical modification on histone. For example, the orange one here, we call it histone acylation. This kind of epigenetic modification affects gene FXN expression. So to test that, and one main character of the epigenetic is reversible. That means this kind of modification can be added and removed from the gene. So we are asking, could we reverse this kind of epigenetic modification in the Friedreich ataxia patient cell lines to the health condition? If that works, could we rescue FXN expression? To test that, we use our epigenetic editing tool. First is dCas9 type 1, which can remove the DNA methylation on gene FXN. And also we test another tool, DCPF1 P300, which can add a histone acylation on gene FXN. We test these two editing tools in the Friedreich ataxia derived iPSC and also in the iPSC derived sensory neuron. We want to see how it works. Here is our result on the right. We can say after demethylation editing, the gene FXN expression level increased roughly 1.7-fold change compared with the patient cell line. And more excitingly, we found the histone acetylation editing can both FXN expression level higher. As you can see here, compared with the gray bar as a control, we can increase the FXN expression level to 2.7-fold. So now we are working to combine these two tools together, and we are going to further bolster the FXN expression level. And in the future, we wish this epigenetic editing tool can be tested in vivo, and hopefully in the future, the epigenetic editing can be used in clinical therapeutic treatment and benefit more patients. That's all my talk. Thank you. Yes, the first question. Yes, we did use TET1 to demethylation the DM hypermethylation region in FXN. We did saw the demethylation decrease compared with the patient group, and we also saw the FXN was increased.
Davy, I think you were on mute. That's why we couldn't hear you.
Apologies. So did you hear the question? No.
Sorry, I didn't hear it.
So yeah, the question was, have you been able to see the direct if TET1 treatment reduces DNA methylation in the FXN gene?
Yeah, the question, yeah, I saw that question. Yeah, after the TET editing, we did saw the DNA methylation decrease and the gene FXN was rescued.
Can you explain maybe to us a little bit more what these editors are? What are they made of?
Yeah. I afraid the time not enough, so I didn't to talk more details. This epigenetic editing tool have two components. One is CRISPR-Cas9, which can specific targeting the gene we want to editing. For example, here we want to specific targeting FXN. We don't want to affect the nearby gene or the father gene, which may cause some side effect. Meanwhile, this Cas9 fusion with some enzyme, which can achieve the writing and remove this epigenetic modification, such as to remove the DNA methylation and also the histone acetylation.
Thank you.
See one next question coming in. Very cool strategy. Have you tested for off-target effects of your effector fusion proteins or other genes non-specifically upregulated?
Yeah, that's a very important question. Every time when we use epigenetic editing tool, this is the first thing we have to test the off-target and targeting efficiency. We test for the DNA methylation, we test the genome-wide DNA methylation change, and we also evaluate the whole genome, the transcript change. And also for the histone acetylation, we evaluate based on the ChIP-seq and the gene expression, the transcriptome profile to evaluate if it affects some gene, well, I mean, like off target cause this gene upregulated or downregulated. This is still under ongoing. So in our previous testing use this tool, the off target effect is very limited.
I see another question. Did you see gene activation in both proliferating stem cells and also in differentiated cell types? And which one?
Yes, we for the DNA methylation editing, so far we only tested in the iPSC it works, and for the histone acetylation, we test both in iPSC and the sensory neuron. We test both in the mechanoreceptor and the nociceptor.
Okay. I have no further open questions at the moment.
I have a question, if that's okay. I couldn't type in here, but I was just wondering what degree of change in acetylation can lead to, 2.5, 2.7-fold of protein increase.
You mean this one?
Yes.
Yes, this is the DPFM P200 for the histone isolation. The gray, there you can see there are blue bar and the gray bar here. The blue bar is treat with dots, which means to activate this editing tool. Without dots means this editing tool was silent, so it's used as a control. You can see compare with the without dot treatment, without activate this editing tool, we almost got 2.7 for the increase of FXN.
And does that correlate to 2.7-fold increase in acetylation?
We will assume so because we quantified the isolation change in this gene, and also we didn't find the off target, so we would assume. And also we have the control here, which is the control for the editing tool, which don't have any targeting. So compared with that, we saw this gene increase. So we will assume it's contribute to the histone isolation editing. Mm-hmm.
Okay, perhaps one final question I see here in the Q&A. Do the iPSCs come from patients with short or long triplet expansions?
Yeah, we test in two pair of patient iPSC. One is the homogeneous expansion of JA, roughly 700 JA repeat. The other is heterogeneous. It's one allele have like 1,300 JA repeat. The other allele is a healthy allele.
You, Shannon. See no further questions in the Q&A. In the chat, not sure if that's okay. Yes.
Yep.
All right.
Next.
So our next presenter is Poire Yamiego from UT Southwestern. So Poire is going to talk about a potential therapeutic approach for FA patients with point mutations. Poire, floor is yours.
So thank you for the introduction. My name is Puire Yamiego. I am a postdoc fellow in the Ampere Lab at the University of Texas Southwestern. I am glad to share our work, a potential therapy for FA patient with point mutation. This is a DNA, and after the transcription, we have, yeah.
Do you want to put it in presentation mode? Perfect. Thank you.
Thank you. Okay. So I'm talking about this DNA, and after the transcription, we have the pre-mRNA, which is composed by the exon and intron. And the intron are removed and exon joined together to form the mRNA. And the mRNA need to be translated to the protein. The process who remove the intron is called splicing. And a good splicing, it's a capital to have a normal protein. The splicing is done by some protein. This is blue, it's a splicing factor. This is a splicing activator, and a red a splicing repressor. But all work together to have a good splicing. And they need to recognize some specific region on the mRNA to do their activity. So a question, what happens if we have a mutation in the splicing factor region? So, and what can be the solution? Let's find the answers in my presentation. We receive a patient with biopsy patient, and we isolate the cell. Everybody have 2 copies of frataxin gene in his body. So the patient have 1 copy with 700 GA repeat, and in the other copy, he have a point mutation in the splicing slide, and his protein, frataxin protein, was very low compared to the control one. When we analyze the RNA of the patient, we found that the splicing is not done in the normal place, but in the aberrant place, and give an aberrant transcript. So we propose to use antisense oligonucleotide to solve the problem. Antisense oligonucleotide ASO, it's a short nucleic acid that can be used to target the pre-mRNA and modulate the splicing. So we design some ASOs and we target the region of the splicing, the regulators. And we found the increasing of frataxin protein in patient cells with the treatment with ASO 82. So to prove that is not a random induction, we made this is a copy of the gene of the frataxin, and we have made two. One without the mutation, wild type, and another with the mutation like we found in the patient. This is called minigene. And we have here a flag, make our minigene frataxin more larger than the endogen one. We introduce this different minigene in the cells. And after the days, we analyze the cells frataxin. This is a frataxin coming from the cells. And this is a frataxin coming from the minigene. And you can see here an overexpression with the wild type. But the frataxin was very reduced with the mutated minigene. But when we had the ASOS 82, the treatment, we see that the frataxin is increased. This confirms that our treatment is very specific to our copy of point mutation. So the answer is yes, the mutation affects normal splicing of the frataxin, and ASO are able to increase frataxin level. So our strategy can be used for FA patient with other point mutation resulting in splicing defect. That's my presentation. Thank you for your attention. Thank you, FARA, for the support.
At this moment, no question in Q&A. Here's one. Is this considered gene editing? Is this something that can be considered in the near future?
It's not a gene editing because our ASOs don't target the DNA. We target the result of the DNA, the transcription. So people think it's not a really a gene editing. The future?
Next question. Nice work. Is this aberrant splicing site also used at low levels in people who don't have this particular splice variant?
Will you repeat, please? I didn't get it.
So is this aberrant splicing site also used at low levels in people who don't have this particular splice variant?
No, the patient who don't have the variant have the splicing done between the exon 1 and the intron here. But this specific patient, we found that the splicing is done here. So it's why we try to solve it, because we think that the point mutation prevent the binding of the splicing effectors. It's not the same in the patient with GAA repeat.
I have a question so you talked about the specific mutations. What about other point mutation? Are there other point mutation where you can use the same strategy?
So there are a lot of mechanisms that we can use ASOS to do. This is specifically for the splicing. But for example, with the GAA repeat, we found that we have an aberrant transcript with the patient with the GAA repeat. We can also use the ASOS to bind where this particular aberrant frataxin ETT is produced. So we have many mechanisms also. We can use also ASO to bind in the polyadenylation site. So there are many mechanisms. We use it in our lab, and we just show the particular way of this point mutation, but we have many mechanisms we can use. Thank you, Lisa.
I see another question popping up. Great work. Now, does your strategy reverse biogenetic deficiencies or FRDA patient cells?
Didn't get, please. Will you repeat, please?
So, does your strategy reverse biogenetic deficiencies or FRDA patient cells?
He's asking if you look downstream of frataxin expression, if you rescue the defect the cells have with the oligos.
So we have the increasing in the mRNA level, we have around 2 or 3-fold. It's not a complete reduce, but we increase sensitively the frataxin level. So, but we have around this level, the increasing. But we think if we have about half, there can be the sufficient for the patient to live because 50% compared to the control cell of frataxin, it's okay. The heterozygote don't have any problem. Thank you.
If there are any other questions, please put them in Q&A. At the moment, other questions for other presenters, perhaps? I don't know.
All right. If there are no more questions, then I'll just thank the speakers today. Everybody did a wonderful job. And of course, our moderator, David and Michaela, thank you so much for doing this. And thank you all for listening. Thank you for also participating in the research that these and other research are doing. By donating your blood, donating your cells, you see how your cells that you donated have been used. Also by participating in clinical trials and clinical studies. And we'll see you in 2 weeks. The next session is May 30th. It will be the second and last session of this year Flash Talks. So thank you and have a good rest of the day.